so many discoveries, for sure. So many adventures.
Yes.
So much science.
Oh,
So
yeah,
much
true.
science. We learned a lot, taught a lot.
Yeah.
Yeah.
And taught a lot.
Yes.
Yeah, let's not forget about that, for the
Hershey and
kids.
Chase.
Doing the rap.
Yes.
Yes.
Oh my goodness.
That was so much fun.
That was very fun.
Yeah.
Oh, and the show, the game show.
Oh yeah, the nucleus and the no nucleus.
Yes.
Are you smarter than a Boffin?
That's
Yes.
right, that's right.
And then Terminator. Let's not forget about Terminator.
Wow.
Or structure and order.
Yes. Oh, the E. coli.
True.
Wow, that was devastating.
Yeah. Yeah, E. coli,
Yes,
Yeah.
yes, yes.
Do you guys think we have any more lessons to teach these kids?
I don't know. I think we're almost out of stuff to teach.
I think there's one more.
Oh, that's right.
Yeah.
There is one more. Wow.
Okay.
Who's ready to learn the last lesson?
I guess they're all.
Are you all ready?
Yes, I'm ready.
Let's get to learning.
Hey, this is Dr. Prahalad. I have something special to teach you guys. I'm sure you guys do recognize this. You see this? 5' to 3', 5' to 3'. Yes, it is the DNA. So let's talk about the prokaryotic DNA. Prokaryotic DNA, 5' to 3' is the coding strand, and 3' to 5' is the template strand. And this is also called the sensing strand, and this is the anti-sensing strand. And do you see the 1+ region right there? That's the promoter region, promoter site. The promoter region for the eukaryotes would start from anywhere between -10 to -35. And there is a small region between the -10 and the +1 region that's called the 5' UTR, which stands for untranslated region. And then there is also another region right here, right after the stop codon, which is the 3' UTR, which is also an untranslated region. So this 5' UTR and the 3' UTR would not get translated once the mRNA sequence has been done. Okay? And now there comes the RBS region, which is the ribosomal binding site. So this is where the ribosome comes and attaches and starts to translate into mRNA. So the 1' region. Now you may ask, what is this 1+ region? What's its function? So this 1+ region is where the transcription starts. It is the first nucleotide that is synthesized by the RNA polymerase. The RBS region is also known as Shine-Delgado region. The area between the 1+1 region and the start codon is where the ribosomal binding site occurs. Now, what is the -35 and to -10 region? These are the sites where the RNA polymerase coupled with sigma factor will bind to. Specifically, if we are talking about the -10 promoter region, this is Tata box. It's also called Tata box because the A and T nucleotides are rich in this region. So DNA is a double strand, as we know, and if you want to access the 1+ site, the sigma factor and the RNA polymerase binds to the Tata-rich region, which is the -10 promoter region, and it is not sufficient to close in, which means the torsional stress is brought upon and it can cleave and start to replicate all these. So the region from +1 to the stop codon is called the open reading frame. It is the amino acid reading sequence of a gene. So ORF is always downstream from the promoter, and promoter is always upstream to the ORF region. So this 1+ region, the promoter site, has to be experimentally designed every single time. And the translation begins at the ATG site, which is from here. The region you see here, right before the 3' UTR, is the stop codon. So it is also called as transcription termination site. So this is where the RNA polymerase, once the RNA polymerase reaches the stop codon, it knows that it needs to stop transcription. So when RNA polymerase approaches the stop codon region, the stop codon kind of signals the RNA polymerase to stop transcription. So this site is also known as transcription termination site. Okay, now that we have gone through the whole transcription sequence, let's go over a few details about what's the difference between eukaryotic and prokaryotic transcription translation differences. So let's take E. coli as an example. So before that, I just want to point out that all RNAs, that is the mRNA, tRNA, and rRNA, are produced in transcription. So the main difference between the prokaryotic and eukaryotic RNAs are there are no introns in prokaryotic RNAs. They are ready to be translated as they are produced. And also there is no 5' methyl G-cap and also no poly A tail in the prokaryotic RNA. The eukaryotic have these because these are the DNA, the mRNA is transcribed in the nucleus and has to leave the nucleus and come to cytoplasm to get translated so that it can produce protein. The prokaryotic is already is done, everything is done in the cytoplasm, so there is no need of 5' methyl G-cap and poly A tail in the mRNA. And then also the third difference is that the prokaryotic mRNA can be polycistronic. So another key difference is that some prokaryotes have an mRNA which can be polycistronic, which means it can synthesize multiple messages at the same time, and some are monocystonic. So it's usually that eukaryotic DNA are monocistronic. And another extremely important difference from a eukaryote to prokaryote is in prokaryotes, the transcription and translation happens contemporaneously, which means it happens at the same time. So RNA polymerase works on the DNA and produces mRNA, and as soon as it is made, the ribosomes come and translate it into protein, which means there is no delay. So you may ask, what is a polyribosome? A polyribosome produces multiple protein at the same time, which is nothing but making the proteins contemporaneously. If a bacterial cells need a particular gene, it is going to make them right away and produces protein. These are the key differences between eukaryotic and prokaryotic mRNA transcription and translation. There are many other major details that you guys need to know about prokaryotic translation and transcription. My colleague Dr. Cameron is going to take over from here, okay? Take care.
Hello! As you just heard from my colleague, wonderful things are about to come your way with what I'm about to tell you about. So we're going to talk about sigma factors, and we're also going to talk about both types of termination that occur with transcription. So I'm going to put on my thinking glasses so that I can think a little bit harder. Oh, I can already feel the brain juice flowing. Okay, so for starters, I want to talk to you about sigma factors, which is a small protein subunit of RNA polymerase that is attracted to promoter sequences. So different types of sequences include consensus sequences, which is a common nucleotide that falls within the protein, and the one that, or rather, if you have a consensus sequence in your protein, it's most likely, or at least it is most efficient in transcribing. So also there's different promoters that have different consensus sequences. Initiation of bacterial transcription. RNA polymerase is including the sigma factor, will bind to a promoter region. This is known as a closed complex. The Tata box, which my colleague previously talked about, the -10 nucleotide, unwinds RNA polymerase, and that's known as an open complex. So, at this point, the polymerase have access to the +1 nucleotide and begin to base pair. So, the sigma factor is then released and it goes and gets another RNA polymerase. At that point, the RNA polymerase slides away from the promoter and begins elongation. So, kind of in an explanation form, if this were to be in more simplified terms, you can think of a parent and a child in a shopping mall. Child being sigma factor, parent being RNA polymerase. Child is bringing the parent to the aisle that they want to get something on. Say that parent doesn't want to get them that stuff, whatever it is, maybe Barbie, soap, whatever they want. Then that child, the sigma factor, goes and gets their next parent, which is the RNA polymerase as well, and they then work out that issue. So, ribosomes then come in and bind and start to make a protein immediately as soon as this happens. And that then goes into what Pari, my colleague, was speaking about. So, I discussed the closed complex and the open complex with you, and I discussed what sigma factors are, which is a protein that assists and guides the RNA polymerase. So at this point, I want to explain termination to you. There's two types of termination. We have row-dependent termination and row-independent termination. Row-dependent termination, as previously explained in the termination video, was when we have a mRNA strand, we have a rut site, and we have a row protein assembled on that rut site. Once assembled, the row protein is going to work its way down the mRNA strand. Once reaching the RNA polymerase, is going to cleave the whole strand from it. However, there is also a stem loop that is involved in this strand. This is a GC-rich stem loop. So it needs the rut and the row protein to then further make it weaker and cleave away from the RNA polymerase. Now I'm going to explain row-independent termination. This is when we do not have a row and we do not have a rut. So, how does it do it? Well, we're going to make a stem loop yet again, but this time we're going to use an AU-rich region. Now, the difference between a GC-rich region and an AU-rich region is the GC bonds are triple hydrogen bonded, which means they're stronger than the AUs, which are only double hydrogen bonded. Therefore, when we're in row independent, it is easier to cleave the mRNA strand away from the RNA polymerase due to the fact that the strand alone in this little stem loop is weaker than the other stem loop in DNA and RNA in row dependent. Excuse me, I have so much going on in my brain right now, all this science. So, hopefully that has been more helpful to you, and now that you can understand row dependent and row independent, maybe you'll get a better understanding. Maybe, who knows, one day you'll join us here at Boffin. Well, that was really helpful to me.
Yeah.
I learned a lot.
Definitely.
Yeah.
It refreshed all the memories.
Yes. Wow. Well, I think that was our last video.
Our very last one.
It's been such a good time.
I
It's
know.
been very fun.
I know.
Very fun.
Yeah.
Oh, what a good series.
Yeah.
Definitely.
Definitely on my favorite.
Yeah.
Yes.
You know it.
Wow, that was wonderful. Well, we hope that you've learned a lot from the Boffin girls, you know,
Yeah.
and maybe next time you'll get to be one of the Boffin girls. Who knows? Stay tuned.